So, Phil, you've sort of highlighted the complexity of the virus itself in its ability to affect multiple species. But every year we have to go get revaccinated for flu because of its change. And on the swine side, it's oftentimes the same practitioners are producing new vaccines, autogenous vaccines, trying to proactively address the isolates or strains that are in their specific herd. Can you maybe talk a little bit about antigenic shift and drift and how and why flu changes so rapidly?
Sure. So, we talked about the different subtypes that are in circulation, and some of those will infect particular species, I guess, in a greater predilection than others. But even within the individual subtypes, you can have a high level of genetic diversity with any of the influenza subtypes itself. And what happens there and what causes that diversity is just pretty much the inherent nature of the virus. As far as it being an RNA virus, and when they go through the replication process, they're naturally prone to have mistakes when they're making a copy of the original virus that's replicating. And those mistakes in their genome can lead to, if they accumulate to be a high enough amount, can accumulate to create a different protein. Particularly, we're concerned about the surface glycoproteins, which are the immunogenic proteins, which are what the targets are for the vaccines. So just the fact that it's an RNA virus going through replication, you can get point mutations that may lead to some genetic diversity. And then influenzas are a segmented virus. And so they're made up of 8 non-contiguous segments. If you have 2 viruses infecting the same cell at the same time, as they're going through the process of replication, which gets pretty detailed, they can swap those different segments, and sometimes they might even swap and change their surface glycoprotein, and then you can get a much different virus. So it is a challenge for both on the human side and certainly in our production livestock side to stay current with vaccines that we know are going to be effective. And the most effective vaccines are the ones that are most closely matched to what's circulating at the farm level, or obviously on the case of people at the human level, and that can be difficult to predict. So our veterinarians that are very well versed in understanding how flu works, they're looking and monitoring very closely what's circulating at a farm, monitoring that diversity and trying to keep pace with updating their vaccines when necessary so they can maximize the control. And that definitely is a challenge for multiple reasons, but it definitely is one of probably the biggest reason that there's so much surveillance and monitoring going on the swine side for influenza.
Yeah. Maybe you can also talked about the monitoring Fluture and what the counterpart is on the human side where they monitor.
Well, because influenza continues to become so much more complex, especially from the genetic side and trying to keep pace and understand the different, what we call clades, or the different viruses within each subtype circulating. Iowa State is fortunate that because we receive so many samples for surveillance, we get a lot of requests to sequence those viruses, and it's become very easy to sequence different components of a virus very quickly and to be very accurate as well. And so we have a lot of that genetic information about influenza viruses circulating in pigs within our database at the VDL, and that doesn't always get into public databases for multiple reasons, but one thing we thought would be useful for veterinarians and producers is to have a public website, although all the information that's contained within there is kept confidential. But what it would present is just the genetic data that we have, and what it can show you is the different frequency of detection of these different genetic clades that we talk about within particularly what we're talking about is the H1 and the H3 subtype. And so it can show you the different changes in the frequency that these different strains, genetic strains of the virus, will kind of rise and fall. What would be the most common ones, and then they would really represent probably the viruses that are the most risk at infecting a production system. And so we can keep track of that. Among other things, there's always some emerging influenza viruses. Some of those actually come from people, and they spill over from people into the pigs. So then you can easily recognize when you have something that's much different.
Yeah.
Or we have pigs that come from Canada and other regions, and different geographic regions will have their own predominance of a particular strain. And those do move with the pigs. And so when they maybe move long distances with the pigs into a particular area, If it's a successful virus, that particular strain might become established as well. And so it just further highlights the complexity that flu can present and why it can be such a challenge. On the human side, they're sequencing probably any influenza that gets detected, and those get sent to the CDC. They're sequencing those. They've got also websites available, one called Flu View, in which you can monitor which subtypes are circulating. You can look if there's variant influenza viruses circulating in people, and those might represent zoonotic infections. So they keep very close track of that as well. So I think between that and now with our Fluture website that we host here at Iowa State, it just helps get a little bit more information out there into the public as far as what's circulating to try to keep better track of what's going on in the flu world on the pig side.